Good morning, Agriculture 10, Introduction to Animal Science. This is our lecture about cell and DNA. Why are we going to study cells? Cells are extremely important. Cells make tissues, tissues make organs, organs make systems, and obviously we put those systems together and we have our animal. The cell function is complex, and we are going to try and understand the basics of our cell and our cell organelles. So hopefully all of you did your worksheet from earlier this week and you drew a cell and you made a list and some definitions of all the cell organelles. There's two basic kinds of cells we're going to talk about. Primarily we're going to talk about eukaryotic cells. Those are complex cells. They have nucleuses. The DNA is contained in the nucleuses. We have other organelles that are rolling in their own functions. Like the mitochondria, the endoplasmic reticulum, the ribosome. Then we have the prokaryotic cell. The prokaryotic cell is a simple cell. It's like a bacteria cell. No contained DNA, nucleus, one long strand, very simple type cells. For the most part, the only place we're going to find this in a upper level animal is in the stomach system of our ruminant animals. Some pictures of some prokaryotic cells. You can see they can grow and replicate quite quickly. We will talk about our prokaryotics mostly when we're dealing with health and disease, working on antibiotics, trying to figure out which antibiotics are going to be the most effective for the specific bacteria that we have. These are free-living organisms. They live independently. So we can culture them, we can stain them. They usually are gram-negative, gram-positive. Once we get those results back, we can then identify the antibiotic that we have that will be the most effective. Just to give you an idea of how small they are, this is a chart. You can kind of put some reference from the egg all the way down to the virus in terms of size. Just a contrast between the animal cell and the plant cell. We're going to spend some time talking about the difference, but we're not going to focus on it. We are going to focus on the animal cell. Our eukaryotic cell in our mammals, hopefully you have all these parts listed and defined already. The plant cell, some of the big differences. The large vacuole, obviously we don't have that in our animal cells. The cell wall is very rigid. That's what helps give it its structures. We also don't have that in our animal cell. And the chlorophyll. So when we start looking at animals, we are not going to work with photosynthesis. That is with plants and chloroplast, chlorophyll. Some of the organelle functions. We have the nucleus. Obviously, that is the brain center of the cell. It contains the DNA. The DNA is what codes for everything that cell is going to do. That's where the chromosomes are. That's where the DNA is. That is the brain center. The ribosome. Those of you that have talked to me before about genetic cells, you know the ribosome is my favorite. The ribosome, it's going to attach to the messenger RNA strand once it leaks out of the nucleus. And it reads it, codon by codon, identifying for different amino acids and building a polypeptide chain. We will talk a lot more about the ribosome and the ribosome function when we work on protein synthesis. Just the basics, right? Genes code for proteins. Proteins are cause and effect. So proteins express our genetics. So our DNA sends out the messenger RNA strand. Our ribosome attaches to it. It reads it. It decodes it in a group of amino acids, which then get to go to the endoplasmic reticulum. So protein synthesis. This is a quick rundown, just like I had explained, where we can't send the DNA out of the nucleus. It has to stay in there and protect it. But we can make a copy of it, and we can send it out of the nucleus into the cytoplasm, where we have to have a decoder, and that decoder is the ribosome. So the ribosome will read it, codon by codon, and it will distinguish it till it hits the stop codon, and then we have a group of amino acids, which we call a polypeptide chain, which heads to the endoplasmic reticulum. The endoplasmic reticulum is going to modify this group of amino acids and start the process into a protein and making it biologically active. Once we get through the process of the endoplasmic reticulum, we have to ship it to where it needs to go in the cell, and that's the Golgi body. So the Golgi body works like UPS. It's going to finish the job of having it prepared to do what it needs to, package it, and ship it. The cytoskeleton is for the shape of the cell and for movement. Mitochondria, that is the powerhouse. So when we talk about the mitochondria, we're going to be talking about cell and muscle energy, ATP, adenosine triphosphates. So we're going to use some oxygen in this process. We're also going to use glucose in this process. So we start out with glycolysis. We move into the mitochondria where we go through the Krebs cycle and we create ATPs for the cell to function. So from the food we eat, we break it down into calories. Those calories usually are broken down into glucose. We go through glycolysis. We start prepping some ATP so that we can run that engine that we call the mitochondria through the Krebs cycle and then the electron transport chain to yield our 36 ATPs. Just to break down, some of you like to see it. We eat, we digest, we get amino acids, fats, sugars, and we absorb them into the bloodstream. Obviously, the amino acids we're going to use in protein synthesis. Obviously, the fats and sugars are going to be broken down into simple sugars like glucose, and they will go into that energy-making machine and building ATPs. This is the energy cycle. You can see that photosynthesis is there from the sunlight into plants. Plants then are going to get harvested and eaten by our animals. Our animals are going to break those down into our amino acids and sugars.